in Ischemic Stroke Trial Results and Impact Analysis. My name is Jeannie Luciano. I am the Senior Manager for Get With the Guidelines Stroke Program. Thank you for joining us today. You are in for a treat. We have two of very elite nurse researchers joining us today. So next slide, please. So before we get started, number one, I want to make sure people understand that we are supported by the Get With the Guidelines program, which is a suite of quality improvement programs for the American Heart Association. Next slide, please. I will introduce our speaker. So our first speaker is Brenda Johnson. Brenda is an acute neurovascular advanced nurse practitioner. She also serves as an assistant director of the Comprehensive Stroke Center at Johns Hopkins Hospital and as associate professor of clinical neurology at Johns Hopkins University School of Medicine. Dr. Johnson holds nursing degrees from Auburn University, Mercy College, and Pace University, as well as a Doctor of Nursing Practice degree from the University of Alabama at Birmingham. She pursued additional training through the Advanced Practice Neurovascular Fellowship in Stroke at Arizona State University. Dr. Johnson is currently actively involved in professional organizations such as the American Heart Association's Cardiovascular Stroke Nursing Symposium, the Maryland State Quality Improvement Committee, and the American Association of Neuroscience Nurses. Dr. Johnson serves as a consultant for the Johns Hopkins Global Center for Organized Stroke Care and develops stroke programs domestically and internationally. She has presented her work at national and international conferences with research interests focused on stroke prevention and health care disparities. Our next speaker will be Debbie Summers. Debbie Summers works at St. Luke's Hospital in Kansas City, Missouri in the Neuroscience Research Department. Debbie coordinated the development of Kansas City St. Luke's stroke program and served as the stroke program coordinator for over 20 years, from 1993 to 2014. And in 2014, she took the role to lead all neuroscience research for the hospital. Debbie is a nursing leader impacting acute stroke care management as she speaks at national conferences and through publication of nursing and medical guidelines. Debbie is active with the American Association of Neuroscience Nurses Organization and the American Stroke Association and has served in leadership roles and on numerous committees. Debbie's honors include the 2011 Stroke Council Award, the American Heart Association 2010 CVN Excellence in Practice Award, and in 2029, the CVN Stroke Manuscript of the Year Award. In 2013, Debbie and members of the Stroke Nursing Task Force received the American Heart Association's Award of Meritorious Achievement for their work in stroke-related activities that include publication of nursing guidelines and initiation of annual AJ Stroke Nursing Symposium. Next slide, please. So just a disclaimer here, the recommendations and opinions presented by our guest speakers may not represent the official position of the American Heart Association. The materials are used for educational purposes only and do not constitute an endorsement or instruction by the AJASA. And the AJASA does not endorse any product or device. Next slide, please. So before we get started today, I'd like to go over a few items so you know how to participate in today's event. This webinar is being recorded and will be sent via email in 24 hours. There are no CEs associated with this webinar, but you may access the certificate of completion in your follow-up email after the conclusion of this event. You will have the opportunity to submit text questions by typing them in your questions pane of the control panel. You may send in your questions at any time during the presentation. We will collect these and address them during the Q&A session as time allows at the end of today's presentation. If you experience any technical issues during the presentation, most can be resolved by refreshing your browser. If your issue is not resolved following the refresh, please refer to the GoToWebinar customer service team, whose contact information can be found in your webinar confirmation email. So thank you so much for joining us today. I'm handing the floor to Dr. Brenda Johnson. Brenda, the floor is yours.
Thank you. Hi, good morning, everyone, and thanks so much for joining us in this webinar today. I want to start by saying I have no disclosures, nor does Debbie. Study funding was provided by the National Health and Medical Research Council of Australia, the Medical Research Future Fund of Australia, New South Wales Health Investigator Development Grant, the University of New South Wales Medicine, non-communicable diseases theme, early mid-career, research seed grant to scheme, and Genentech for the USA. So just to give you a little background on, how this study came about, I'm going to go through the timeline and just a few other slides to just give you the full context of why we decided to do this. So this slide demonstrates nursing monitoring after IV thrombolysis in acute ischemic stroke patients. Despite all of the advanced treatments in stroke care and IV therapy, intravenous thrombolysis is the only applicable and approved medical treatment since 1995. Standard care of post-IV thrombolysis requires a lot of monitoring of neurological status and vital signs due to the increased risk of ICH. One thing that we do know about this is that it's very disruptive to the sleep of patients and may impair recovery. So prior to our study conducted at Johns Hopkins, OPTIMIST in 2020, and OPTIMISTmain that was conducted in 2025, the paper that we're going to discuss here today, the intensive monitoring was based on the NIN stroke study. And you're all familiar with the neurochecks and vital sign assessments for that study. So how do we come up with OPTIMISTmain? So in February 2017, Debbie had a discussion with Dr. Craig Anderson from Australia on a feasibility study to monitor frequency of vital signs and neurochecks in patients that had mild to moderate stroke. So what happened after that? We did a lit review. And within that lit review, we looked at thrombolytics and ICH, we looked at the NIH stroke scale and ICH, we also reviewed NIH stroke scale and critical care needs, just looking at the patient population to see if we would have any hemorrhages, and also to look at the critical care needs that might come up with this patient population. So finally, in OPTIMIST, the initial OPTIMIST trial in January 2014 was published in clinicaltrials.gov. Debbie came to me and she said, Brenda, I ran across you guys' paper in clinicaltrials.gov. I saw that you had a small pilot study, and she contacted Victor, Rudy, and I, and we set up a face-to-face meeting to discuss this. And we had that discussion sometimes around 2018 at ISC. And so in October of 18, we reviewed the Johns Hopkins protocol with everyone that was in that meeting. We identified prominent U.S. sites and international site participants. Debbie and I put our heads together and just sent information out by way of emails to all the coordinators and everyone that we knew. And then in February 19, we had a steering committee dinner at ISC, and we got there to talk about the most important thing. We have this great idea, how are we going to fund this study? So we discussed funding. And then a little bit later, we organized a breakfast at ISC. And in that breakfast, we communicated again by way of email. We had 33 sites that were interested. We had a total of, I think, about 42 people that attended, that showed interest in the study, and we're like, oh wow, this is great, we might be getting somewhere. There's nothing better than nurses and nurse coordinators' input on this low intensity monitoring study. So just this slide is just going to describe a little bit. I'm going to describe a little bit about our implementation timeline. So we did get funding, as they said, for Genentech for 25 sites in the United States. In February 2020, the steering committee met again at ISC, and then guess what? COVID happened. I'm like, oh man, this is slowing everything down, right? So we finally ended up getting IRB approval around December of 20, and we started enrolling patients. The U.S. enrollment was completed in December 23. We enrolled about 1,386 patients. And then if you look at the blue bar down here, this just shows enrollment for all the participants in all the different countries. It occurred between April 21 and 24. So it was a very multidisciplinary, not multidisciplinary, but a very international study is the word I'm looking for. So the safety of low intensity monitoring. So we had this great idea, we got people involved. So, okay, we really, really want to know if this is safe. We thought about the trial and we wanted to ensure that a non-inferiority trial was feasible, right? So first, we decided on this non-inferiority design. We did a lit review, and in that lit review, we looked at the NINS trial cohort. The symptomatic ICH rate we found was higher with TPA compared with placebo. Placebo 6.4% versus 0.6%, and this was in the NINS trial. And then we looked at other large studies, or registries of post-approval use of TPA in stroke patients that demonstrated, reassuringly lower rates of symptomatic ICH, 2% to 7%. And so, we said, oh, this is great. So we knew that the second thing that we wanted to do was to compare the new treatment with an active control study, which was standard monitoring versus low intensity monitoring. So the risk of ICH transformation, so my slide says the meeting has ended. Oh my goodness, did you guys see that? I'm sorry. So the risk of ICH transformation post-thrombolysis is another thing that we looked at. And we know that the NIH stroke scale is the most robust factor that's associated with symptomatic ICH risk. So we looked at a pool analysis of almost 7,000 patients from multiple trials comparing TPA with placebo. And the absolute risk of fatal ICH related to TPA increased from 1.6% with a baseline NIH score of 5 to 10. All righty. So next slide. So with this, are you guys still seeing the correct screen? I'm having a few technical.
We are. We're seeing safety of low intensity monitoring.
Okay, it just kind of blacked out a little bit over here. So this was the safety of low intensity monitoring. So this was a lit review from the NINS TPA original trial study group, and it was based on hours from the start of TPA and the NIH stroke scale. As the score increased, the percentage of ICH increased, right? So you can look over here, you see the NIH stroke scale from 0 to 10. You saw that the percentage of ICH or hemorrhages were very low, but as the scores got higher, the percentage of bleeding increased also. And if you pivot over here to the non-fatal by hours from the start of TPA, the highest death was in the earliest hours, like within the first 2 hours. We did not change monitoring, in the initial study until after. We did not change monitoring in OPTIMISTmain until 2 hours. All right. Does that make sense? Yeah. Okay. So this was a paper. This slide shows the predictor of critical care needs and the rate of intervention based on the NIH stroke scale. This is a paper we published at Johns Hopkins. In our paper, we chose a population that was less likely to hemorrhage with an NIH of less than 10. So it's safe to monitor these patients in a non-critical care area. This is why, we chose those patients, because if you pivot over here and look, you see as the NIH stroke scale went up, you saw that there was more of a need for critical care beds or critical care needs. So patients with an NIH above 10 had a 7.7 times higher risk of requiring ICU resources as compared to patients who presented with an NIH of less than 10. Most patients with ICU needs developed them prior to the end of TPA infusion, and only 7% of patients without ICU needs by the end of the TPA infusion went on to require ICU care later on. So the aim of our study was to determine safety and efficacy of the Johns Hopkins low intensity monitoring protocol in patients with acute ischemic stroke of mild to moderate severity. And those are patients, again, with an NIH of less than 10 within 2 hours of the end of their IV thrombolysis and who do not require critical care needs. The hypothesis we compare, as compared to standard high intensity monitoring, low intensity monitoring is non-inferior for functional recovery at 90 days. And we hypothesize that it's safe, cost effective, and more acceptable to nurses and patients. So the low intensity monitoring here, you guys are not familiar. The standard monitoring, you're very familiar with these time frames. This is the low intensity monitoring intervention. You know, every 15 minutes for 2 hours from the start of the IV thrombolytics, then every 2 hours for 8 hours, then every 4 hours for 14 hours. So the OPTIMISTmain study design, this was an international pragmatic step wedge cluster randomized control and non-inferiority trial. There were 4 steps and there were 3 different groups. As you can see the 4 steps here, and there were 3 different groups. The randomization was computer generated. It was a computer generated step wedge process. So we had nothing to do with that. We let the computer do that work. Every site started at the standard. You know, so you can see by here that every site started at the standard level. What else was I gonna say here? Let's go over this. So yeah, and so data collection occurred at 0 days, 24 days, and 7 days. And if they were discharged, it occurred a little bit earlier. It was a blinded assessment because a study nurse could not communicate with the patient. Right here, all sites received standard nursing and medical care, so we didn't deviate from the medical or the standard care. So all of that was fine. And one thing that I did want to mention, I know that's where my thought process was with Debbie. So what I was going to say, I just remembered that every site started as standard with the step wedge, but Debbie, I'm calling you out here, was really mad because her team did standard 3 times, whereas other teams went to step 1 and then they went to the standard. So sorry about that, Debbie, but I wanted to share that with everyone, and I'm sure that people on the call experienced the same thing as well.
It was my team was upset.
Okay, clarification, Debbie's team was upset. The IRB allowed us to do the waiver of consent because there's currently insufficient level 1 evidence specifying the appropriate level of monitoring. The intervention, you know, can be considered low risk on the basis of the pilot data and insufficient evidence to indicate there is substantial harm associated with less intense monitoring. Patient care will not be compromised by either standard or less intense monitoring. Consent at the individual level had the potential to introduce selection and responder bias and compromise recruitment and the feasibility of the study. So let's talk a little bit more about the consent process. So patients were informed of the study within 24 hours, but you know, by then they would have already received the intervention. We consented the site and not the individual patient. So waiver of the consent was built into the design study, because if we had consented every individual, patients would just be at an increased risk of bias, you know, and that's why we consented the sites. By adopting the protocol, we made sure all patients with an NIH of less than 10 and ICU requirements, you know, received the protocol, got placed on the protocol. But here in the US, we used a waiver of consent, but internationally in other countries, there was an administrative consent that was done. And what I mean by that is that the hospital CEO had to sign, you know, had to sign the consent, had to sign for the process to enroll the patient if they agreed to it. So it was a little bit different internationally versus in the US. This is the opt-out consent. So we also had an opt-out. Patients were given an information sheet the first 24 hours, and on that information sheet, it stated the hospitals participating in research that is assessing standard nursing practice to post-thrombolytic stroke care around the world, and the need for data collection in hospital and 90-day follow-up assessment was explained to everyone. So with the opt-out, it meant that they do not, well, the patients didn't want data collected that is put in the case study report. We put it in the case study report, and you know, we did not contact the patients at 90 days. Therefore, data was not collected at all for this population. So what is non-inferiority? It's a randomized trial to assess if a new treatment is no worse than a standard treatment by a predetermined margin. OPTIMISTmain is a clinical study that demonstrated low intensity monitoring was not worse than standard monitoring by a small predefined non-inferiority margin. We chose the lower boundary of the 95% confidence interval, which was 1.15, for the risk ratio to define a non-inferiority margin. And so if you look at the slide here below, I put this in because, you know, non-inferiority trials are becoming really, really popular in stroke. So this is the non-inferiority margin, the dotted line. And so you can see that, you know, by this bullet, you know, B, it's non-inferior, but it doesn't cross over this non-inferiority margin. You can see to the left of this is harm, to the right of this is benefit. And so our trial was not, you know, non-inferior, right? And so it did not cross over the line, you know, the non-inferiority margin. So we, you know, this was done, I mean, the way we came up with this, you know, was by this non-inferiority trial was that we looked at prior randomized controlled studies and, you know, and consensus among the clinicians in our study. So we had to have consensus among the PIs and other clinicians in the study. We compared the low intensity monitoring to standard, and we just set our non-inferiority margins. Clinicians, you know, had to determine if there was a slight change in the efficacy of the study. You know, they had to think about that if we wanted to go ahead and pursue that. If there was a slight change, they also looked to see if we had a potential for benefits of this study, right? So things, what I mean by benefits is such things as, you know, less sleep interruption, you know, less nursing resources. So I'm gonna pause here and let Debbie go into more criteria about the enrollment, and then we'll recap at the end and talk a little bit more about the non-inferiority margin, sharing, you know, it specifically to our study. This is just an example that I threw in here to explain the non-inferiority trial. Debbie?
Okay, we're going to talk about OPTIMISTmain enrollment next. Advance the slide, Brenda. Okay, when we developed the clinical trial, we looked at the inclusion criteria as any trial would have. It was pretty straightforward. You had adults with the age of 18 or older. They had to have a diagnosis of acute ischemic stroke that had been given TPA or TNK. They had to be clinically stable. And what do I mean by clinical stable? Their vital signs had to be stable. There was no need to be taken frequency vitals because of IV infusions or blood pressure management. These patients did not have any airway compromise or need ventilatory support, because you couldn't let someone stay without checking them more frequently. You looked at their medical history. If there was any suspicion or possible, they had a medical history that required monitoring them closer, you could not put those patients in it. You didn't need ICU admission. Now, my hospital continued to admit to an ICU. Brenda's hospital used a step-down type progressive care unit for those patients, as many of you probably do out there. But they didn't need, we just didn't have another spot to put them. They were not needing ICU care. And then the clinician had to be comfortable with the low intensive monitoring. So every PI or every patient or physician who gave TPA, you know, we had to say to them, are you okay with us putting the patient on that? Next slide. The exclusion, it was kind of like a double, the way we said it twice, but the exclusion, the patients were not medically stable. And we put some examples in the protocol. So if they post-TPA needed a continuous IV infusion, we did define if they had to have 3 or more doses of labetalol or they had to have IV hydroxyzine at the dose of 10 or higher within that 2 hours, then that would not be a patient that you wanted to put into that trial. And you couldn't put a patient in if you were needing to put them on pressors and vasotension because of hypotension. The other thing, which you guys have probably all of you have seen a patient who you raise the head of the bed and they quit talking, their arm quits moving. So they were perfusion dependent. So we couldn't put that patient in there. We could not use, do a patient that was requiring BiPAP or high flow oxygenation or someone that was intubated. And then if you had during that first 2 hours or at any time during that 24-hour, if you had a change in the NIH of 4 or more points, then any of these that would happen, you needed to, by looking at the above criteria, even though they put on the OPTIMIST monitoring of low intensity, if there was a change in that patient, you had to return them to standard vitals and neural assessment during that initial 24 hours. Next slide. We developed a resource tool because these patients, so this is a low intensity monitoring algorithm. Other centers use this. So the patient would get TPA, that's at time 0. It wasn't till at 2 hours that you made the determination if this patient could go into the trial. So I just created, because it was the nurses at the ICU or the ER that were looking at this patient at 2 hours, if the NIH stroke scale and were they medical stable. So you went down. If the NIH stroke scale is less than 10, then yes, they could continue. But if that NIH stroke scale was greater than 10, that stopped it. They were not a candidate. So when we look at medical stable, if they were on a blood pressure drip, they were on either one, increased blood pressure, decrease it, that stopped it. But if you did assume that they were medically stable, then you would say, yes, they could be placed. And we would talk to the physician and say, this is a patient that can go on the trial. We chose at our site not to put endovascular thrombectomy patients on because of the frequency monitoring of groin. And we knew we'd be in there and we could have a bias, but that was a site decision. Other sites put their patients on it. Next slide. How we implemented this then is we just added our research vital sign, the OPTIMIST trial, the low intensity monitored on our ischemic stroke patient management orders. So the physician had to check one, is this a patient that was a standard of care and needed the regular frequency, or were they safe to go to the low intensity, and that would be the OPTIMIST patient. So during that first 24 hours, we also emphasized anytime there was any change in the patient, you returned back to the standard of care. And then we had to kind of remind nurses in our team that once you put them into you couldn't move them from standard of care to low intensity. So example, you had an NIH of 10 at 2 hours. And at 3 hours, the NIH was 9. And they would call us and say, hey, can we put this patient now into the OPTIMIST trial? And we had to say no. The criteria is what was the NIH at 2 hours. Sorry, you got to continue on with standard of care. Our hospital now has, with the paper release, and we have adopted the low intensity monitoring. It doesn't say for research now, it just says the patients do standard of care or the patients do low intensity monitoring. So our advanced practice nurses, our neurologists, by the time they get to the ICU, the TPA's been delivered. It's right about that 2 hour, and they will mark which vitals use. Next slide, please. So let's look at the analysis of our enrollment. So you'll see on the left the countries. I think it's pretty cool. I never thought I'd be involved in an international trial. The USA started first. We were pretty slow because COVID stopped us and IRBs got slowed down. but I will tell you, Chile, particularly Australia, some of these others talk about rigid. They allowed no research to be done. We got ours completed a lot, like a year almost before some of the trials or some of the sites were able to get up and down. So the blue was the standard monitoring. The red was the intervention. So we had the control versus the intervention. I know it's very slow or very small, but you can see that all the sites that did standard versus intervention. And if we look at, Brenda has explained the cluster, there were actually 181 hospital sites through this whole study that were assessed, are they eligible? There was certain criteria. You had to have a research department. You had to have someone that was going to be a champion of this trial, like you guys know. You had to have a designated place that these patients went. You couldn't have a patient go to 14 different places in the hospital, like some of the smaller. So we started out with 181. 61 sites were excluded, and then we were put into 120 clusters that we randomized into 3 groups. So like in the US, we had 25 sites, and 7, 7, 7, 8, 8, 8 were put into the different clusters. And I'm not going to go over that. It talks about the average size of those clusters. We did have 1 hospital withdraw, and 1 was not activated. So that brought us down to 118 hospitals. And then 4 hospitals did not recruit. So out of all the hospitals, there were 114 hospitals that recruited. And I think it's very important, we screened 9,092 patients. Almost 9,100 patients were screened. That's a large number of patients that were screened. Next slide. So we had to exclude 4,170 because they didn't meet medical stability, or the NIH was too high at the end of 2 hours. There was all those reasons. So totally, we had 4,922 patients that were randomly assigned. They were assigned either to low intensity or the standard monitoring. 2,789 low intensity, 2,133. We had some loss to follow-up. We were missing primary outcomes. The main primary outcome we were missing was that 90-day not modified Rankin. You guys who are involved in research, you know that is such a pain sometimes trying to get a hold of someone at 90 days. But we had put into the trial, we knew that we would have about a 10% loss to follow-up. So ultimately, at the end of the trial, we had 2,559 that had data. And in the control, the standard, we had the control, 1866. So next slide. So let's look at when you set these non-inferiority margins, you set our protocol, you develop the protocol, then you enter all the data, and then begins the analysis of your data. So one of the things that we wanted to make sure is, did we have monitoring adherence? So what that means is, did we take was the monitoring frequency, did we adhere to that? So did we adhere to the protocol? And we had great adherence in this. And that was because nurses took the role on this study. This may have been written by some clinicians and physicians, but it was you guys at the bedside who really took charge of these patients. So we had 93% and 94%. So there was no difference in the adherence. As far as the median duration of how long we monitored, it was 25 versus 24 hours. We wanted to make sure that the median neurological assessments and vital assessments were correct. So for a low intensive monitoring, you only need to do it 17 times. For a standard monitoring, it was 37 times, because it was difficult for nurses sometimes to go, oh, but I haven't checked them for 4 hours. Yes, you did check them. You were in that room. You were monitoring them. It's not like you left them for 4 hours and didn't go evaluate them. You monitored them, you just don't take the vitals except the designated according to that protocol. And then we looked at the monitoring location. We did this just to kind of see, because throughout, even before this study, many of you have been putting the TPA patients in step-down areas. Internationally, they call them high dependency units or immediate care, acute stroke units. I just have an ICU. But if you looked at the location where these patients were, and we looked at where they all went, because this is data we collected, you will see that there was no statistical significance and that it was safe to go to the other areas. Next slide. We also looked at all the variables. We had to record when we collected data. Were they intubated? Did they have an NG, a G2? Were they mobilized by a therapist? Did they have TCDs? Because we wanted to make sure, we promised in this study that every patient would get the standard of medical guideline and nursing care. So we looked at that. Did everyone get managed the same? And between the low intensive monitoring and the standard monitoring, there was no difference. In the ICU, we had more patients, less patients that went to the ICU. That's an important. I've been begging at my hospital to have like a step-down unit. It would be safe. And talk about resources. Talk about when my neuro ICU is totally full and you've got a patient with an NIH of 2, they don't need the ICU. So this could be used in working with a management that patients who went to the ICU did very well. There were fewer that went to the ICU in our study compared to the standard monitoring. Next slide. Our primary outcome was a modified Rankin at 90 days. You guys know modified Rankin is a functional outcome. Our primary outcome that we set was a 0 to 1. And 2 to 6 was considered not as well, not functional. So when we looked at this and looking at it, the modified Rankin really had no change. When we look at 0 from low intensity to standard monitoring, same with, none of this was statistically significant. The problem, and I'll show it in another slide, is we chose a very high, a very strict modified Rankin as our primary, 0 to 1. Those of you who are involved in stroke trials, 0 to 2 is usually acceptable. We wanted to make sure this study, it was stringent and we had patients that we were, would be safe, we wouldn't affect, any patient care or any decline. So we set ourselves for a really high modified Rankin of 0 to 1 was our acceptable primary outcome. Next slide. We looked at secondary outcomes. I'm not going to go over all these. I'll talk about some highlighted. But you can see to the left, we looked at everything. We looked at, NIH stroke score, their sleep, patient reported experience, how long they were in the, before they discharged, death. You look at symptomatic hemorrhage, but that wasn't because of the monitoring anything. We chose, Brenda went over, we chose the proper patients that weren't going to be risk of hemorrhage because we chose patients with low NIH. So if they did an analysis because our outcome did not meet, we had weak evidence of being non-inferior. So we didn't exactly totally meet the non-inferior. If we would have chosen the modified Rankin score of 3 to 6 and only looked at 3 to 6, that was a non-inferior trial. We also looked at the patients like a modified Rankin of 3 to 5 without death. We looked at death at 90 days. But since we chose our primary outcome as 2 to 6, we were not non-inferior. So I'll show you the weak evidence, but it was a good trial, but that was not harmful. And these showed no clinically significance in the outcomes of these two standard monitoring groups, or two monitoring groups. Then we analyzed all the subgroups. I'm not going to go into each one of these because you notice, look at it, that plot, that data point right along the standard of care. If it had gone far to the left, that would be low intensity monitoring was better. Far to the right, the standarding was better. We stayed right in the middle. It's just that we touched the line of our non-inferiority, but we didn't meet non-inferiority. Next slide. The strengths of this study is that it was very complex. It was a complex study for implementation in the real world. There would be many times that you could have taken extra vitals, or that you could have not followed the adherence, and then that would, but the strength showed that the monitoring adherence was very good. It was a very robust, it had patient-centered outcomes like NIH stroke scale and death, and were they on ventilators? So everything looked at, and we had multiple countries. We went from high income to low income to middle income in all of our countries. And this showed this was a large sample size. The results were generalizable in these countries and in this ethnic and social demographic diverse population. The inclusion was broad. You had to be 18 or older. You had to have an NIH stroke scale of less than 10 at 2 hours. You couldn't be critically stable, but every patient got standard care. And we had a pre-specified, the Georgia Institute in Australia, there were many statisticians that worked on this study, and we knew exactly what was going to be analyzed. And I cannot go into Cox and all the different analytical plans, but we had it all set up. You had to define your confidence in or your non-inferiority margin. If we would have done the study results, then we could have defined it, we could have set it, but you can't do that. You set your non-inferiority margin first, and then you do all the analysis. Next slide. So limitations, it is a step wedge. That is complex. You have a potential for data quality issues because of practice changes. I mean, we really I had to keep saying to my nurses, this patient is, and I actually had to write a deviation up because we took one patient and just started measuring all the vitals we wanted to on him because she was in there doing other things. So I had to really say, this patient is on low intensive monitoring. The other things that occurred, some of the hospitals already started doing low intensity during COVID. They were in survival mode. So those hospitals, I think Ohio was one of them, they had to go back to standard because they'd already been doing it. So nurses are, okay, why can't we do it? The other limitations is because of COVID and our low enrollment, we had to increase the number of hospitals because there was low patient. We actually wanted a indication of a 90% power, but we had to recalculate to 80% power. It was an open trial. As a study, I had people who admit the patient. I didn't see the patient, or they would consent them or talk to them about it. I typically did a 90-day follow-up call. So I didn't know anything about the patient. So that's the reason it was blinded. Potentially confounding effects of just background care. There was none evident. Some hospitals used, I already said this, some hospitals used ICU, some used step down, some used, so we had all, but there was no evidence of, causing problems. And then every study, you have to say, is it really generalizable? Can be a limitation. Next slide. So as we wrap up, in conclusion, OPTIMISTmain provides weak level evidence. Remember, I told you we did not meet our primary care modified Rankin of 0 to 1. So we had weak level evidence that low intensity monitoring is non-inferior to standard monitoring in patients with level of neurological impairment, mild to moderate. So we had patients under NIH less than 10, so that's the mild to moderate that we chose, and we showed no harm with these patients at all. It was no clinical meaningful differences in these patients, and the outcomes were not bad. There was no harm at all. So there was strong evidence for safety and functional outcome because of no change in the groups. And if you look at the end, OPTIMISTmain is safe. It did not seem to cause any harm to approximately 5,000 patients. That's a large clinical trial. The low intensity monitor was safe. It was well accepted by the patients, the nurses, clinicians, and it will likely could produce cost savings by the lower ICU bed use. We've got another, one of the statisticians is writing a paper on, there were several questions. The international team asked patients what they thought about not being woke up so much. We didn't do that in the US. And they also did sleep analysis. So our recommendation is hospitals should consider incorporating this approach to enhance systems of care according to local circumstances and resources. Next slide. Publication was released. We got to speak on this at Finland at the European Stroke Council, and the publication was released that day that we spoke. Next slide. We want to acknowledge there was a steering committee, there was all kinds of statisticians, safety board, patients, families. Next slide. You can go to the next two slides. Those are just our references, so if you want to look at more. And thank you so much for joining us. And Jeannie, we'll turn it over for questions.
Okay, wow. So great work gets done when you follow through on insightful nursing questions, right? And just as a note, if you want to be proud of nurses, just look to these two women. Nurses get to impact lives of patients every day. So we have a lot of questions here. We're going to get through as many as we can. So I guess this is a site who thinks they could have been interested in doing a study. How did you get the information to sites that you were starting this study so that they could demonstrate interest?
It was many times by, we had emails out, Genentech reps were talking to sites. We could only choose 25 sites, and there was so many beyond 25 that wanted to be a part of the study. We sent emails, we had flyers at ISC, that ISC breakfast.
Yeah.
That there was probably organized in special invitation, because then when Genentech said they would only give us funding for 25, we had to stop.
Yeah. And it was also shared, you know, by way of listservs. You said that already, but, you know.
Yeah, I put it on AANN. I think we had it on AHA. There was a lot of listservs. Yeah.
Yes.
Fantastic. How did you handle the language barriers with the multi-country studies?
You know, multi-country speak English better than I do probably sometimes. With our steering committee, everyone that was on the steering committee spoke English, and then each country had their nurse advocate. I know Dr. Urati is working with Malaysia, I believe it is, or working with one of the countries. And when Brenda and I were on a committee with him, or on a Zoom call not too long ago, she spoke English to us, or she had an interpreter with her.
Yeah, a lot of countries had bilingual people that were spearheading this study. Yeah. And all of the international participants that were on the steering committee or participated were bilingual.
Yes.
There's a question about what was the monitoring frequency after the Q4 hours times 14 hours?
Well, it would be post 24 hours, and you would do according to what that patient needed. If you were in the ICU, you might do it every 2. We didn't set anything beyond the 24 hours.
That's correct.
It was patient indication and the treating clinician.
Correct. And I want to add to that, Debbie, it was based on the unit protocols.
Yes,
You know,
yes,
if they're in a step down unit, if they're in an ICU unit,
yes.
the protocols vary.
Okay, there's a question here, and I think I know this answer, but is there a resource you can recommend that verifies the care of a patient can be monitored post-TPA by a step-down nurse and does not have to be a critical care nurse? Is it not like the level of the nurse, it's really
No.
the in the unit?
Well, you would want to have that nursing group trained. They would need to be trained. Brenda presented the safety of putting a patient into a step-down unit based on the Ischemic Stroke scale. You would have to have a process that chose the correct patient that could safely go to that step-down unit. You would want to train your nurses. They'd have to be NIH certified. They'd have to be, trained to look for complications. You'd have to have the order set that gave them. Any other questions, Brenda, or did I answer from your thoughts, Jean?
I think I'm just making sure that I understood that question because, most of the people that were participating in the study were trained. We had monthly sessions, Debbie, if you remember, we had monthly sessions with coordinators and, we handled Q&As like this. A lot of different questions came up like this.
But I think they're asking now, like they want to take their hospital and give them a patient and step down now. Can a step down nurse take that patient versus a critical care nurse?
Oh, absolutely. Yeah. I mean,
if they're trained.
Yeah.
Okay. So, did any of the research sites use telestroke? This person says, we usually don't keep our TNK patients because we don't have any on-site neurology. They appreciate your input.
So you are what we used to call a drip and ship, or they're a site where they give T&K and then they ship the patients. We have small hospitals that transfer a lot of our TPA patients to this. We have not changed any protocol. They need to do frequent monitoring after they give the TPA at the small hospital and then educate their EMS or Lifelike group to do frequent monitoring until they get to our hospital. Those patients we weren't able to put into the trial because most of the time it was beyond the 2 hour. I don't know if I'm answering the question.
I think that does answer their question. There is a question here. Did you have required baseline modified Rankin scale for enrollment?
No. We
So
just
not a pre-morbid modified Rankin.
No, there was no exclusion as a pre-morbid. We had a pre-morbid modified Rankin that we recorded, and then a 90-day. But there was not an exclusion criteria for baseline, like most clinical trials, because this wasn't a drug trial. It wasn't like you don't. A lot of our clinical trials have an exclusion if your modified Rankin is greater than 2.
There's a question. Yeah. There's a question here about, is there a graphic about it, maybe in the journal article, is there a graphic about how far you missed the non-inferiority margin by?
Yes. Yes. It's like figure 3, memory serves me correctly. I'm not sure, but it is in the paper.
We came so close. We were so close. Let me see. It is figure 3 and figure 4.
Yes.
Thank you. And where there's a question about where were the results published?
They were published in Lancet. And you can go to the Lancet Journal. It was
So I
published
can pull up
online.
that slide again.
Yeah, pull up that slide.
Here we go.
But we didn't put the date on there. It was published May 21st, 2025.
Perfect. So here's a question. It's a great presentation and study. We have been informed by several Joint Commission reviewers that it would be premature to implement the results from this study. Do you have any updates on this or suggestions for hospital and would like to implement results from the study? So how do the certifying bodies, or do you know how the certifying bodies feel about it?
Well, that was interesting because my Joint Commission reviewers said, I am so glad you guys are doing this. I'm sure yours did too, Brenda.
Yeah, absolutely. There was a
They raved and said, why aren't people, but that's the unfortunate discrepancy or, in the reviewers. We didn't have any problem.
Yeah, I didn't either.
Reviewers specific there.
I haven't heard that.
Say that if you're going to incorporate this into your program, I would list it as one of the clinical practice guidelines, or I would list it, somewhere, in your protocols, your policies. It has to be listed.
One more question, and we'll have to log off here, but is data of patients who ended up not having a confirmed acute ischemic stroke included in the study?
Yeah, on one of the graphs, and there is a stroke. We looked at the stroke mimic, and I want to say the stroke mimic was, because we could go deep into all these graphs. I want to say it was 10% for both groups, because yes, let me see.
I was trying to find it. Yeah, I think you're right, Debbie.
I think it was 10%. So we kept them in it. And it was good to look at stroke mimic, which we all know that stroke mimic is about 10%. It did not change with low intensity or high intensity.
I was going to say, I think that's consistent with a lot of clinical trials.
Oh, yes, yes.
Well, I cannot thank our presenters enough today. I want to thank everyone for attending today's webinar. We appreciate the time that you take. We also appreciate the care that you are giving to improve the outcomes of patients with stroke in your communities. Once you leave today's webinar, you will receive a survey on the presentation. We would appreciate it if you would complete that and provide your feedback. You will also receive a follow-up email within 24 to 48 hours with a link to view a recording of today's webinar. So on behalf of our presenters and the American Heart Association, we want to thank you for joining us and have a great rest of your day. Thank you, everyone. Take care.
Jeannie, we put our emails there. I think Brenda and I both welcome emails if we can help in any way.
I can confirm that they are very receptive to sharing their information. So thank you. Thank you, Debbie. Thank you, Brenda.
Thanks.